Hello students, this video is about photosynthesis. By the end of this video, we should understand the process of synthesizing organic molecules using energy from the sun just a little bit better. To begin with, let's look at the overall equation that's used in photosynthesis. We're going to use energy from the sun as our power source for this. We're going to take 6 carbon dioxide molecules and 6 water molecules, and out of the process we're going to get a glucose molecule and 6 oxygen molecules. Keep this equation in the back of your mind as we go forward. First, let's take a look at some organisms that do photosynthesis. So we immediately think about green plants, which do in fact do photosynthesis, but there's a little bit more diversity of things that also do photosynthesis. For example, we have a brown kelp. It photosynthesizes. This right here is a single-celled euglena, a paramecia. It also photosynthesizes, as do algae. There's many things in nature that are going to use this process. Let's specifically look at a plant, a green plant, because they're the ones that we are most familiar with in doing photosynthesis. If I were to take a leaf and I were to do a cross-section of it, I would see the top. I'm going to see a bunch of cells on the inside called mesophyll. At the bottom, I'm going to see these little things called stomata. So stomata are basically holes in the leaf, but they're able to close down. When they close down, there's not going to be any transfer between the inside of the leaf and the outside. So the reason they might close down is because when it's really hot, transpiration, just how water evaporates, might happen really, really quickly, and the plants don't want to dry out. And so when it's really hot, or it's really dry, or it's really windy, stomata may close. When those stomata close, then carbon dioxide can't go in and oxygen can't come out, and so that might actually slow the process of photosynthesis. When photosynthesis is happening though, if we look at a single mesophyll cell, on the inside we're going to see a bunch of these structures called chloroplasts. Let's look at the part of a chloroplast. It has 2 outer membranes, outer membrane and an inner membrane, with a little bit of space in between. Most importantly we've got these little stacks of pancakes. So each one of those pancakes is called a thylakoid. If you have a whole stack of pancakes, it's called a granum, and if you have multiple stacks, they're called grana. Then there's all this goo inside the chloroplast, and that's called the stroma. You should immediately notice a lot of similarities between a chloroplast and our good old friend, a mitochondria. Inside that chloroplast, the reason they look green is because they have a pigment called chlorophyll. So chlorophyll is going to be really important in absorbing the right photons of light to allow photosynthesis to happen. It can't, however, absorb all photons of light. So if we remember, light is on the electromagnetic spectrum. We've got infrared over on this direction, our visible light here, and ultraviolet light on this direction. Within our visible light, there are different wavelengths that represent different colors. Chlorophyll looks green to us because if we have green right here, the green light is going to hit that chlorophyll, bounce off, and go into our eyes. So it's not going to be absorbed by the chlorophyll itself. Versus if we had another color, like blue, blue is absorbed by that chlorophyll. This allows us to look at how different types of light can impact the growth and the ability to do photosynthesis in plants. There's a couple different types of chlorophyll. Chlorophyll A, chlorophyll B. We also have some additional pigments that can do photosynthesis as well. We notice that chlorophyll A is going to have this peak amount of activity over here near the violet. Chlorophyll B is going to have a peak amount of activity in blue. Notice the amount of activity, the amount that is absorbed, drops to near 0 at green. That's why it looks green, but green light will not be helpful in photosynthesis. Then as we get more towards our red side, we see that the activity increases again. The reason to keep this in the back of your mind is because plants can't grow in just green light because they can't use that light for the steps of photosynthesis. So let's take a look at those steps. There's only 2 big steps in photosynthesis. We have first the light reactions. We also have, secondly, the Calvin cycle. Calvin cycle is also called sometimes the dark reactions or light-independent reactions. There's a couple different names for all of these things. Notice our overall equation. We have light coming in, water coming in, carbon dioxide coming in, and then our final products are oxygen and sugar. Notice this thing is all happening inside the chloroplast. Let's break down these steps just a little bit more. We'll start here with the light reactions. Notice these light reactions are happening in the thylakoids, so they're being done across a membrane. Generally, when we have a membrane, that's going to allow us to create a difference, a concentration gradient, and that's the big thing that's going to happen in the light reactions. So these images represent what's going on during the light reactions. So there's a couple big things that are going to happen. First, the light reactions are going to produce some ATP. Second, the light reactions are going to produce NADPH. So NADPH is an electron carrier. In cellular respiration, we had FAD2, or FADH2, and we also had NADH. Here we just have NADPH. It works in a similar way, just has a different name because it has a slightly different chemical structure. So in order to do this, we're going to start here with light. Light's going to come in, it's going to hit something called a photosystem. Photosystem 2 happens first because it was discovered first. There's not a good reason that photosystem 2 is the first one. But light is going to hit this. As a photon of light hits this, it's going to excite two electrons. Two electrons are going to jump up and be accepted by another protein that's embedded in the membrane. Then that's going to be passed down to the next protein, and passed to the next protein, and passed to the next protein. And as the electrons are passed between proteins, that is going to allow some hydrogen ions, some protons, to be pumped on one side of the membrane into that thylakoid on the inside. We're eventually going to get to photosystem 2, where another beam of light, another photon, is going to re-excite those same electrons so they can again be brought back down here and eventually accepted by NADP+ to create NADPH. So all of this has to do with the movement of electrons. So the first question is, where do those electrons come from? And the answer to that is water. So water is there. Water is going to be split. As water is split, it's split into its protons, it's split into the electrons, and then oxygen is left over. So oxygen will be produced in this step as a byproduct. The plants don't actually need it, it's just there because they split the water. Let's look at that another way. So if we're looking at this image here, this is the inside of the thylakoid, this is the stroma on the outside of, like inside the chloroplast. And we see again, we've got light coming in, and every time the electron moves between these different proteins, these hydrogen ions, or these protons, are going to be pumped on one direction into the thylakoid. Once there's a whole bunch of hydrogen ions on one side, they can flow back through ATP synthase, just like in cellular respiration, and create ATP. So overall, we know transfer of electrons here is coupled with pumping of hydrogen ions and the splitting of water molecules. We know that the big things that are produced here are ATP and NADPH. So that's the light-dependent reactions. Once we have ATP and NADPH, those two things are going to go to the second step, where eventually we're going to get glucose out. Another way to think of that first step is kind of like this diagram. So you start by exciting an electron. That photon's kind of like hitting a mallet, getting that electron to go up. And then as that electron is passed down, it's going to pump hydrogen ions on one side, which is going to provide the concentration gradient to make the ATP. When it's re-excited, it's going to allow that to happen again. Eventually, the electrons are collected by NADPH at the end. As you're looking at this, you should be thinking about just how amazing it is that the structure of the chloroplast allows this to happen. It requires a membrane in order to do all this process, and just look at how much membrane there is in the structure of a chloroplast. Every thylakoid is just a section of membrane. Also notice the similarities between a chloroplast and a mitochondria. Mitochondria has all these folds to allow for a bunch of area, and chloroplast also has all these different pieces of membrane. In our mitochondria, the inner membrane space is going to be where the hydrogen builds up. It's going to be in the thylakoid space in chloroplasts. Then the hydrogen ions, the protons, are going to flow back through to create ATP into the stroma of chloroplasts, this gooey stuff, or in the matrix, this gooey stuff of mitochondria. And so these two structures, though different with very different roles, super similar in some ways as well. Again, we can think of it this way, where you just have thylakoid space where lots of hydrogens are building up as electrons are being passed down, and ATP is eventually going to be made. So here we are with NADPH that's made, with ATP that's made. Next step is to go to the Calvin cycle. Calvin cycle occurs in the stroma. So Calvin cycle, like its name, is a cycle. It's going to repeat over and over again. First, you're going to add 3 carbon dioxides to the cycle. Then it's going to go through a bunch of different steps. As it goes through these different steps, ATP is going to be required, NADPH is going to be required. Eventually, we're going to get half of a sugar. Half of a sugar is called G3P, and then this cycle goes around twice to allow a glucose to be put together. The one thing you'll hear a lot about with this, there's a bunch of intermediate steps, but Rubisco is going to be that first step, so sometimes you'll hear that. If you hear Rubisco, just think Calvin cycle. So again, things that are coming in, carbon dioxide, ATP, and NADPH. Things that are coming out, eventually a sugar, and then ADP and NADP plus get recycled back to the light reactions. So overall, our process allows us to eventually produce glucose, which we could put together to be amino acids, fatty acids, we could change it to be starch, we could eventually get sucrose for export. There can be some additional modifications once we've done this process. There's a couple of things that'll impact the rate of this process. I want to think a little bit about why. First is light intensity. And so we require photons of light to excite those electrons. And as you add more and more light, the process is going to go faster. Eventually, though, all of the proteins in the thylakoid membrane are going to be working, and no matter how much light you add, they can't go any faster. And so the rate of photosynthesis will increase, and then it'll level off. Similar with carbon dioxide, because you require carbon dioxide for the Calvin cycle, but once all the Calvin cycle is working in the stroma, you can't go any faster. And so as you add more carbon dioxide, rate of photosynthesis increases and then levels off. You can, however, change the rate of photosynthesis to a peak rate if you're looking at things like temperature or pH. And that's because this whole process is done with enzymes. And so everything that impacts enzymes is also going to impact the rates of photosynthesis. For example, if you increase temperature, when it's really slow, it just takes a while for the molecular movement to allow the reactions to occur in the enzymes. Then you'll get to an optimum temperature. If it gets too hot, you denature the enzymes and it won't work anymore. Photosynthesis won't work. We see that with pH as well. There's going to be an optimal pH. There's other things that will impact the rate of photosynthesis as well, but keep these ones in the back of your mind. The last thing I want to leave you with is just a little bit about why we care about photosynthesis. So there's a bunch of different reasons. It produces oxygen, which we use. It produces sucrose or glucose, which we eat and use as fuel. There are 2 related processes between plants and animals. Plants themselves even do cellular respiration and photosynthesis. Photosynthesis is our process of making the molecules that we can store, and then cellular respiration is getting them back out in order to use them. There's this pretty famous experiment done where you had an animal, put him in a sealed bell jar, died. Plant, sealed bell jar, died, but both together, and they were able to live. So think back to ecology. Photosynthesis and cellular respiration work together to allow life. The final thing is I want to encourage you to look at this YouTube link if you have a chance. It's just some guy talking to a bunch of British people about where they think the mass of a tree comes from. It's kind of amusing, so you might want to take a look at that. Okay, so hopefully that's helpful in getting some of the background with photosynthesis. Thanks.